I want to take a couple of, as I noted from the top, our audience questions are more important than mine, so I want to take a few audience questions if it's okay with you guys. Sticking with the target identification theme, Harch would like to know, how clean does a target need to be? How does potency of the payload play into that? Any takers on that one?
I think maybe I can take that one. I think if your target is not clean, you know, I mean, you need to understand where it is expressed, and you need to in your payload selection, you need to have first a rationale and on why your payload MOA would not affect the tissue, the off target. I mean the undesired tissue that express your targets. What's you know, if you go maybe for a tubulin agent, which is supposed to kill replicating cells, and your target is expressed in a tissue that is not growing, maybe there you can get away with a not clean target. You know, that's but you need to have a rationale for that. You need to demonstrate it. You need to de-risk it. You know, you need, so there is this preliminary work that you have to do here, and try to score with that target. Yeah.
Thank you, Léo. Philipp, I'm not going to let you off the hook, because Harch also had a question specifically for you. He's fascinated by your dual payload approach at Araris, and he's wondering if you can speak to what considerations were taken into account in terms of whether the different drugs impacted different stages of the cell cycle.
Yeah, so that's a good question. No, I mean, the different approaches how you choose dual payloads or multi-warhead ADCs, and typically, and sometimes you can also experience surprises which you may not necessarily see in vitro, because we have seen strong synergistic activity in vivo but not in vitro. So, and it depends probably, so a good indication is whether the cancer tissue which you're going for, whether there is an indication if the payloads you're choosing or the mode of action of the payload is actually sort of validated in the indication you go for. So for example, if you choose colorectal cancer as an indication, I mean, it's known that tubulin inhibitors are not so active in those indications, so you may not want to choose a tubulin inhibitor. However, the question is, what happens when you add a topoisomerase 1 inhibitor to the tubulin inhibitor, and you go for colorectal cancer still, because the tissue may behave differently. Because, for example, when you apply topoisomerase 1 inhibitors and you induce DNA damages using topoisomerase 1 inhibitors, the cell uses microtubules to repair the DNA damages. And of course, when you block now with the MMAE, for example, this repair mechanism, you may be able to overcome and induce new, like a mode of action which previously the tissue was not sensitive to. So I think using multi-warhead ADCs, you can really develop novel ADCs that are differentiated compared to others and may open up avenues which, as mentioned, conventional monopayload-based ADCs may not be active to. So I think this is quite an exciting field, and it really depends on, as mentioned, on your imagination, and also, of course, on whether there is data available of a single mode of action. But as mentioned, it's not the end of the story, because there's a lot of biology behind, which you first also need to read about, how these mode of actions actually are working of the single payloads to better understand and design then eventually the ADCs. So,
thank you, Philipp. Miles, let's take a look at the first polling question. We'll ask the audience a question, and then we'll get back to our interrogation of the panel. So this first question is, which of these do you consider the biggest challenge in your ADC development effort? Those of you listening in on the panel, please make a selection now. And while you do, Philipp and Léo and Monika, I'm curious, looking at that list of choices, does anyone venture a guess what will be the leading challenge that our audience faces in ADC development? Monika, you mentioned earlier you mentioned the narrow therapeutic window. Do you think that might be a leader?
Could be. Let's see, what the audience comes up with. But my pick would definitely be a narrow therapeutic index, or even for that matter, tumor penetration, or off-target toxicity. So that is also something which we analyze, and we as pathologists, I tend to look at tissues. So we look at them and we try to analyze the off-target toxicity as well. So I think toxicity also, as we mentioned, earlier, that narrow therapeutic index and off-target toxicity could be also very, very important. And this is where the optimizing the patient pool becomes very important, especially if we look at tissue biomarkers, identifying the patients which are going to respond to that drug. That selection of the patients and optimizing the patient pool becomes important.
Excellent.
But as far as chemistry and conjugation is concerned, I think I will defer the question to Philipp and Léo.
Well, yeah, we're going to get into that real shortly here. Let's take a look at the poll results and see what our audience had to say. Manufacturing complexity. Yeah, so we have some questions on that I want to address too, that I think Philipp and Léo will be happy to respond to. But looks like that and the narrow therapeutic window are our leading challenges. Any reflections on that? Anything surprise you about the poll result there? Philipp, Léo, or Monika?
Yeah, maybe I'll start here. So my personal view is traditionally, when you look back, why many ADCs have failed in clinical development, at least from a clinical development perspective, it was because they had a narrow therapeutic index. You could not typically achieve efficacy without having toxicity, so you had more toxicity than efficacy. That's why actually many ADCs had to stop in clinical development, which was due to the narrow therapeutic index. Now I think there was a learning in the ADC space, and data showed that when you choose a different linker and a different payload, like the HE did, for example, with an HER2, and when you use a payload which is per se much more tolerable, it helps you to achieve a better therapeutic index, because they could go into indication where, for example, Kadcyla, which is a microtubule inhibitor, could not go, and they could really address a much larger patient population compared to Kadcyla. It's the same antibody, it's just a different liquid payload that actually seemed to enable this much broader patient population, or addressing this much broader patient population compared to Kadcyla. Now let's see. And also the tolerability of Enhertu and using ADC, or when you look at ADCs using the same linker payload from the HE, you typically have a very relatively good or high tolerability of between 5 to 6 mg per kg. While when you use a more potent payload, if the same payload class like exatecan, it's a more potent payload class, it actually has a poorer tolerability because the payload is much more potent, and we'll see where that ends up. And to me, it's from, as I mentioned initially, for me it seems like the narrow therapeutic index, it's actually has been a challenge for ADCs because of let's say lack of suitable technology, and also maybe a learning effect that actually has taken place in the ADC space to better develop, or ADCs that have a higher therapeutic index, and to really address more patients that still express the same target compared to approved ADCs.